Plant nutrition encompasses the essential chemical elements and compounds vital for plant development, metabolism, growth, and reproduction, as well as their external supply. Failure to provide these necessary components can hinder a plant's ability to undergo a regular life cycle or form crucial plant constituents and metabolites, aligning with Justus von Liebig's law of the minimum. There are a total of 17 crucial plant nutrients, with elements like carbon, oxygen, and hydrogen being obtained from the air, while others, such as nitrogen, are generally sourced from the soil. Plants must derive mineral nutrients like nitrogen, phosphorus, potassium, calcium, sulfur, magnesium, carbon, hydrogen, and oxygen from their growth medium. Additionally, micronutrients including iron, boron, chlorine, manganese, zinc, copper, molybdenum, and nickel are also essential for plant growth. These nutrients are present in the soil as salts and are absorbed by plants as ions, with macronutrients being consumed in larger quantities. Notably, hydrogen, oxygen, nitrogen, and carbon collectively make up over 95% of a plant's total dry matter biomass. Conversely, micronutrients are found in plant tissue in minute quantities, typically measured in parts per million, ranging from 0.1 to 200 ppm, or less than 0.02% of dry weight. While most soil types worldwide can naturally provide sufficient nutrition for plants adapted to the environment, in cropped soils, it becomes crucial to enhance soil fertility artificially through fertilizer application to stimulate robust growth and maintain or increase yields. Plants primarily obtain carbon, hydrogen, and oxygen from the air and water they absorb. Justus von Liebig's research in 1840 revealed that plants additionally require nitrogen, potassium, and phosphorus for optimal growth. Liebig's law of the minimum states that a plant's development is hampered by any nutrient deficiencies. Arnon and Stout showed in 1939 that molybdenum is crucial for tomato growth when cultivating plants in mediums other than soil. Plants absorb essential elements through their roots from the soil and through their leaves from the air. Nutrient uptake from the soil involves cation exchange, where root hairs release hydrogen ions into the soil, displacing attached cations for root absorption. In the leaves, stomata enable the intake of carbon dioxide for photosynthesis, using the carbon molecules as a carbon source. The root, particularly the root hair, plays a vital role in nutrient uptake, with its structure influencing the rate of absorption. Nutrient ions are transported to the root center, the stele, to reach the conducting tissues, xylem and phloem. The Casparian strip in the root regulates nutrient and water uptake by preventing passive flow of substances. Water and mineral ions are transported by xylem, while phloem facilitates organic molecule movement. Water potential dictates nutrient uptake, with nutrients flowing from areas of higher solute concentration in the soil to regions of lower solute concentration in the plant. Plants uptake nutrients through simple diffusion, facilitated diffusion, and active transport mechanisms. Nutrient mobility within the plant directs resources to younger leaves over older ones, which display symptoms of deficiency first if nutrients are scarce. Symbiotic relationships with microorganisms like rhizobia and mycorrhizal fungi enhance nutrient absorption. Legumes house nitrogen-fixing bacteria, converting atmospheric nitrogen into usable forms, while non-legume plants like wheat and rice rely on nitrogen compounds in the soil for growth, which can be supplied through various sources like fertilizers or decomposition processes. Nitrogen, an essential element for plant growth, is predominantly sourced from the soil despite being abundant in the Earth's atmosphere. Certain plants engage in nitrogen fixation to convert atmospheric nitrogen into a biologically useful form. However, most plants primarily rely on nitrogen already present in the soil. Plants like soybeans, beans, peas, clovers, and alfalfa are known for their ability to fix nitrogen. Conversely, agricultural staples like corn, wheat, oats, barley, and rice need nitrogen compounds in the soil to thrive. While carbon and oxygen are mainly obtained from the air, other nutrients are absorbed from the soil. Fundamental nutrients obtained from air and water play vital roles in plant development. Carbon serves as the building block for various plant molecules, while hydrogen is essential for sugar formation and overall plant structure. Oxygen, acquired in different forms from air and soil water, is integral for both photosynthesis and cellular respiration in plants. Macronutrients like nitrogen, phosphorus, and potassium are crucial for plant growth and development, participating in key metabolic processes and structural components. Nitrogen, a significant component of plant substances, is essential for protoplasm formation and amino acid synthesis. Phosphorus, another vital nutrient, is crucial for nucleic acid structure and various cellular functions. Potassium, although not directly involved in metabolic processes, plays a crucial role in enzyme activity, turgor regulation, and stomatal functioning. It is essential for carbohydrate and protein formation, enzyme activation, and photosynthesis, contributing significantly to plant growth and development. Sulfur, a crucial structural component of various amino acids and vitamins, is essential for chloroplast growth and function. It is particularly vital for the formation of iron-sulfur complexes within the electron transport chains during photosynthesis. Notably, sulfur is indispensable for the process of N2 fixation by legumes and the conversion of nitrate into amino acids, eventually leading to protein synthesis. Moving on to calcium, this macronutrient mainly resides in the leaves of plants, with lower concentrations found in seeds, fruits, and roots. Its primary role lies in being a fundamental element of cell walls. When combined with specific acidic compounds present in the pectins of the middle lamella, calcium forms an insoluble salt. Apart from this, calcium is deeply involved in meristems and plays a crucial role in root development, encompassing functions like cell division, elongation, and hydrogen ion detoxification. Its functions also extend to neutralizing organic acids, inhibiting certain potassium-activated ions, and aiding in nitrogen absorption. A prominent indicator of calcium deficiency in plants is a compromised root system, with roots typically exhibiting symptoms before the above-ground parts. Moving forward to magnesium, its standout role in plant nutrition revolves around being a constituent of the chlorophyll molecule. Serving as a carrier, magnesium partakes in numerous enzyme reactions as a potent activator, closely linked with phosphorus compounds that supply energy. Keep in mind that plants are adept at accumulating most trace elements proficiently, as some serve as sensitive indicators of their chemical surroundings. Barrier mechanisms in certain plants regulate the uptake of specific elements, like how different plant parts accumulate distinct elements based on the surrounding soil and groundwater qualities. Considering micronutrients like iron, it is indispensable for photosynthesis and functions as an enzyme cofactor. The absence of iron can lead to intervenal chlorosis and necrosis, underscoring its importance. Similarly, copper plays a vital role in photosynthesis, enzyme processes, and the synthesis of lignin, markedly impacting grain production. Manganese, crucial for photosynthesis and chloroplast development, can lead to abnormal coloration in foliage when deficient. Sodium, essential for some plant metabolic processes, can substitute potassium's role in regulating stomatal activity and water balance in certain plant types. Lastly, zinc is pivotal for various enzymes in DNA transcription, with zinc deficiency manifesting in stunted leaf growth. Nickel is essential for activating enzymes crucial in nitrogen metabolism and can substitute for zinc and iron in enzyme cofactors. Chlorine aids in osmosis, ionic balance, and photosynthesis, while cobalt has shown benefits in certain plant species, especially aiding in nitrogen fixation. Silicon, though not deemed essential, has been found to enhance plant strength, health, and productivity by improving cell wall strength, root mass, and overall plant vigor. Vanadium, selenium, and vanadium have varying degrees of importance in plant growth, health, and stress tolerance, each contributing uniquely to the overall well-being of the plant kingdom. Mobile nitrogen is transferred through the xylem from the roots to the leaf canopy in various forms such as nitrate ions, amino acids, or amides. It can also move in the phloem sap as amides, amino acids, and urites, allowing mobility within the plant. Older leaves tend to display chlorosis and necrosis sooner than younger leaves due to nutrient transport dynamics within the plant. Phosphorus, being mobile, first displays signs of deficiency in older leaves. Magnesium and potassium, also mobile nutrients, show deficiency symptoms first in older tissues before progressing to younger tissues. Conversely, calcium deficiency manifests in new growth as it is immobile in the phloem and relies on transpiration for supply. Boron remains fixed in the plant and must be provided to growing parts through the xylem. Sulfur cannot be relocated from older leaves to support new growth, leading to deficiency symptoms appearing first in the youngest tissues. These deficiencies can result in yellowing of leaves and stunted growth. Nutrient deficiencies in plants can range from subtle growth rate reduction to severe stunting, deformities, discoloration, distress, and even plant death. Identifying deficiency symptoms can be challenging, as they are often multiple and moderate, with nitrogen frequently being the most limiting nutrient. Macronutrient deficiencies like nitrogen typically lead to stunted growth, slow development, and chlorosis, along with distinct color changes in leaves. Phosphorus deficiencies may resemble nitrogen deficiencies, causing intense green coloration or reddening in leaves. Calcium deficiency can be observed in leaves through curling towards the veins, blackened appearance, and issues like blossom end rot in certain plants. Potassium deficiencies may lead to necrosis, chlorosis between veins, higher pathogen susceptibility, and wilting. Micronutrient deficiencies such as zinc or boron can impact various plant parts, affecting growth and fruiting. Boron deficiency, for example, results in stunted growth and improper cell wall formation. Boron toxicity can occur at concentrations higher than 1 ppm in the soil-water solution, being toxic to most plants. Availability of nutrients like nitrogen, important for growth, is influenced by various factors including microbial processes like nitrogen fixation and weathering of rocks. Phosphorus availability is crucial for plant growth and can be enhanced through relationships with mycorrhizae, especially in soils with limited phosphorus content. Moreover, nutrient interactions and environmental conditions play a significant role in nutrient uptake and plant growth. Calcium and magnesium can inhibit the uptake of trace metals, while zinc can impact iron levels in plants. Imbalanced application of fertilizers can lead to decreased crop yields. Boron availability to plants is influenced by soil pH, with deficiencies appearing during dry weather due to decreased nutrient availability in desiccated soil layers. Nitrogen uptake by plants varies depending on soil conditions, with plants predominantly taking up nitrate ions, especially in environments like boreal forests where ammonium is more prevalent due to nitrification levels. The evaluation of plant nutrient status, known as ionomy and nutrient profile, is often represented through tissue elemental analysis. Interpretation of these findings has stirred controversy in recent years, as mathematical models now supersede the traditional law of minimum by considering interactions among various nutrients. Nutrient elements do not function independently, but rather engage in chemical interactions and affect each other's uptake and biological functions through various mechanisms, illustrated in cases like ammonia. In the realm of agricultural systems, boron's high solubility in borax or boric acid leads to leaching from soil, making it unsuitable for fertilization. Instead, less soluble calcium borate is derived from sodium tetraborate. Boron, often applied incidentally in soil amendments, needs careful monitoring to offset the loss during cropping. Selenium stands out as an essential mineral for animal and human nutrition, vital for preventing deficiencies arising from selenium-poor soils. Introducing inorganic selenium fertilizers can enrich crops and animal diets, thus enhancing animal well-being.